So this video is amino acids backbone, peptide bond formation. I made a typo there. And so basically a peptide is the same thing as an amino acid. Those words can be used interchangeably. And so the peptide bond, this is a type of covalent bond that holds one amino acid to the next. And I'm going to abbreviate amino acid AA. And so this is kind of scary looking chemical reaction up here, but basically all we're doing is really focusing in on this one little bond right here. And here's the vocabulary that should correspond roughly to this video, and here are the SLOs that correspond as well. Okay, so here we have lots of chemical bonds going on here, and basically what I want you to do is kind of break it down, pay attention to what's going on here. If you don't recognize it right away, what we see here is a chemical structure that has an amino group, a carboxyl group, central carbon, and then a hydrogen up here, and then we have another hydrogen down here. So this is, hopefully you would recognize this. Do you know what that is? It's an amino acid. And pop quiz, do you know, is this in a cell? Is it in an aqueous solution or not? So pause the video and think about what that might be. Okay, so it is actually in a cell or aqueous solution. The way we can tell that is because we can see the positive charge on the amino group and the negative charge on the carboxyl group. And so this is one amino acid, also known as a peptide, and then right next to it over here we've got another chemical structure, so let's check that one out. So this, we also have an amino group, a carboxyl group, and a hydrogen, and then down here we have a different chemical kind of pointing down here. So do you guys know what this is? Number one, so what's this functional group? That's the first question. And then the second question is, what part of the amino acid is this? From when we talked about the core features of an amino acid, like which part of it does it symbolize? Okay, and so I'm going to pause the video and answer those questions. Okay, so first question about what functional group it is, CH3, that's a methyl group. And then what part of the amino acid is this? Well, if we can rule out that we've got amino group, carboxyl group, and a hydrogen here, then the last thing it could possibly be coming off that central carbon is the R group. And that means this is a different amino acid than the other one that's over here because it has a different R group. And it would have a different name. I don't know what it is, but we could look it up on a chart. And basically what we're trying to do here is see that these two amino acids are hanging out in the cell, and then they're going to be brought together, and they're going to form a bond between them to form a growing polymer. And so this bond that forms between them is a peptide bond. Remember, we can also call an amino acid a peptide, and when we have lots of amino acids in a row, that's a polypeptide. So basically all those names kind of go together. And so basically what's going to go on here is this is going to be a situation where we're going to make a macromolecule chain. So what's the general name of that reaction? So take a minute and think about it. So what's the chemical reaction that forms a macromolecule or polymer? And so, as a reminder, we are going to do that's called the condensation reaction or dehydration synthesis. And remember, with those names, dehydration synthesis or condensation reaction, that refers to water being removed. And so check out the chemical structures here of the two different monomers that are about to be joined together. through a peptide bond. And notice that they've got a couple atoms that are highlighted in blue. So we've got our hydrogen, oxygen here. And of course that's H2O. And so these are the atoms that are going to be removed that will allow for now the two monomers to be free up space to form a new bond together. So this is water is removed. Notice this is a little different from what we saw on some of the books, the figures in your textbook. So we saw that sometimes they would draw the OH coming off of one monomer and the hydrogen coming off the other. It doesn't matter so much for our purposes, though. I'm sure in chemistry there's a good reason for that. The important thing is that we see that we can balance the reaction here. And so now this carbon is going to form a bond with this nitrogen here. And so the bond that's formed is right here. This is a peptide bond. And so you should be able to recognize the peptide bond, which is this situation here. So obviously the bond itself is just a single line, a carbon bonded to a nitrogen. But typically when we talk about a peptide bond, we also need to think about what that carbon is bound to. And so we consider the context of that. And so this carbon, which is bonded to a nitrogen, also has a double bonded oxygen to it. And so what that means is that if you're given the option to write out what a peptide bond is, you should write it as C bonded to nitrogen, and then that same carbon double bonded to oxygen. And notice that there's a water molecule that's got removed over off to the side here. And so now we've made a just a very short polymer, just 2 units. And some might not call that a polymer because that's not many units, but this is basically the same process that would be used. And so the next monomer, next amino acid that might come in would be over here, and then we'd repeat the process. That then it would be oriented with the amino group to the side, and then we'd pull off this oxygen and the hydrogens here, and we go down the line with that process. And here we can check out that more detailed structure. So this is a little overwhelming to see at first, but notice that we have a real pattern going on here. So basically, first let's check out what's going on the bottom here. So basically we have these green boxes which indicate the monomers, which are amino acids for our polypeptide structure. We've got 1, 2, 3, 4, 5, 6, 7, 8 of those, and we see that one of them on this guy here we have an amino group hanging off, and that's what we call the N terminus. On the other end we have a carboxyl group hanging off, and we call that the C terminus, C for carboxyl, N for the nitrogen and amino. And basically, if we look at the very detailed structure here, we can see that we have our amino acid structure here, first one, second, third, fourth, etc. And so we can count those across nitrogen, carbon, carbon, nitrogen. You don't need to do a lot of this, but the reason I'm trying to point this out is to see why we call it a backbone. So basically, we see the amino acid backbone here. This is the part that is the same across all the amino acids. And then pointing off here, we see the R groups. And notice that those are variable here. And basically, as you know from other lectures, that now these side chains are going to kind of get creative and start to flop around in the aqueous solution, and then they're going to start to form some chemical interactions. And so some chemical interactions are going to last for a longer period of time. Other ones are just going to kind of bounce off because they're not a match, if you will. So you could think about that here. So of these, like who might match together, so maybe pause the video and find 2 things that would be likely to form a chemical interaction, and then also state what type of interaction that would be. Okay, so I'm going to talk about what some of those are. So the first one that I noticed was we've got this guy here and this guy here, each of which have lots of carbons, and so those are likely to form a stable interaction together. So think about what those R groups are classified as. Are they polar, nonpolar, or acidic or basic? And so those are going to be nonpolar. And so what they're going to do is they're going to form hydrophobic interactions. And they could also form van der Waals interactions, which is another type of chemical interaction that occurs with nonpolar amino acids or other nonpolar molecules. Okay, so now pause the video again, see if you can find another possible interaction between the side chains. Okay, so let's check out another one. I'm going to change the colors here. Okay, so if we check out this guy here has the OH group, so this is going to be a polar amino acid. And then is there anybody else on here that's polar? This one would also be polar because it's got an OH. Also might notice that the color coding here is a little bit of a tip-off. And so basically polar and polar can form a stable chemical interaction. And so what is the chemical interaction that could form between them? And we've talked about hydrogen bonds. Okay, so those are some examples looking at this chain of amino acids. As we talked about before, that's primary structure when we look at the chain of amino acids, and now as I'm starting to talk you through interactions between the side chains, that's when we're getting into tertiary structure. Okay, so let's talk a little bit about the protein folding. So basically, what we see here on the lower right is we see this kind of unfolded protein, and so it's kind of got this ribbon structure. This is symbolizing many different amino acids, and we don't see all their chemical structures. And as they go through the process of protein folding, then they can take on these more complex shapes. And so this includes secondary and tertiary structure. And you might see some of each of those here. So can you see any secondary structure within this folded protein? And also, can you see any tertiary structure? So pause the video. Okay, so there is secondary structure here and here and here and here. Each of those would be an alpha helix. And then we can also see that these things have folded back on themselves, and that would be tertiary structure. And this process of protein folding is facilitated by something called a molecular chaperone, which is kind of a cool name. These are other proteins in the cell that help proteins take on their proper shape through folding. This could be new proteins, this could be slightly damaged proteins. Molecular chaperones hang out in the cell, and they just kind of patrol and try to help stabilize the protein shape. And then this part of the figure basically is showing you what our textbook usually draws it as, is just this kind of blobby thing, but this is the reality of what is going on with the shape. Also want to tell you a little bit about this process. So proteins can hang out in this relatively stable state for quite a long time, but just like anything, there's wear and tear on them, and so they can get damaged, and they can kind of fall apart. And so there's a special name for that. So as proteins fall apart, if and when proteins lose their folded shape, they are denatured. So it's a vocabulary word. So this denatured state is implying that it would be natural to be folded, and so it's kind of a weird name, but they're denatured, and so they can't function when they're in that kind of unfolded state. If there are molecular chaperones around, they could potentially refold that protein into the proper shape. There's a lot of different things that can cause denaturation to happen. Heat can cause this, just kind of normal wear and tear in the protein. Ionic changes, like major changes in pH or salts, could also cause this to happen. Okay, so that's the end of this video. Basically what we have seen here is how we can form a polypeptide chain through the process of adding peptide bonds between the amino acids. And we also saw here how we can do some protein folding and some of the bigger picture aspects about what it means for a protein to lose its shape and then how it might regain it.